Hello, everybody. I hope you're still in the mood to learn about proteins. Last lecture was all about structure. Today is all about function. First, we will be categorizing the general functions of proteins, and then we will generalize how protein functionality can be regulated and the physiological benefits of doing so. Today's molecule of the moment is sort of a surprise. I will be asking a random student to draw amino acids for the class, and I'm not going to tell you which ones here because then it wouldn't be a total surprise. So make sure you study up before coming to lecture. Let's do a quick brainstorming activity. What do you think are the main functionalities of proteins? I would pause the video, flip back through your notes, or just think of proteins that you're generally aware of and attempt sorting them into 4 different bins. And I'll give you a moment to consider, and I'm going to go ahead and start writing down my categories. So far, I mentioned on a few occasions proteins such as collagen and viral capsids. Collagen holds our tissues together, capsids hold a virus together. So proteins have a very famous structural role. That is worth discussing. What else is there? Hemoglobin is a very famous protein, which we will be discussing today. What does that do? Sometimes people think it transports iron. It doesn't. It transports oxygen by using iron. Ferritin is a protein in our blood that transports iron. So transport and storage are related protein functions. Ever since 2020, I feel like the whole world obtained a degree in epidemiology and virology. Viruses make you sick, and when you get sick, you make antibodies to detect and destroy those viruses. And antibodies are really great recognition proteins. And related to this, of course, is signaling. Examples include antibodies and antigens, growth factors, hormones, so on and so forth. Lastly, one of my favorites, enzymes, catalysis. Too many to name, but just off the top of my head, digestive enzymes like pepsin, which help break down protein. I don't know, maybe I'm hungry. Altogether, I think these categories cover all bases. I'm sure another biochemist would maybe divide them up differently, but the important thing this exercise is meant to do for you is just to show the breadth of activities proteins are capable of doing thanks to the highly modular nature of primary structure, amino acid side chain, and all the levels of structure that ensue from that. That previous exercise may entice you to ask the question, where do proteins get their functions from? And yes, the answer is their fold, but it's actually so much more than that. Take, for example, post-translational modifications. These are covalent modifications that install or modify a protein's functionality. Proteins can have their functions turned on or off or dialed into a specific intensity after they have folded. And I think you've probably heard of some of these before, such as phosphorylation, the transfer of a phosphate group to usually a regulatory serine residue. Most of the time, occasionally it's threonine and tyrosine as well. Shown over here is phosphoserine, and see how its hydroxyl side chain is able to join with the phosphate anion very nicely. And this is again why it's so important to know our amino acids, to have a visual memory and instinctual memory of them. That's why I keep on harping on this chord, so to speak. Another cool example of a post-translational modification is methylation and acetylation. These typically occur on lysines, specifically on histone proteins, and these modifications are responsible for silencing and activating genes. And this is pretty cool. When you methylate a lysine residue, you quaternize the nitrogen elements, and it's always going to be positively charged, and so it's going to bind the negative DNA very, very tightly, and that's going to keep it scrunched up and not accessible to translation machinery. When you acetylate the lysine's nitrogen group, it is trapped into a neutral electronic state, and that's going to attenuate its affinity for DNA, let it loose, and let that gene get transcribed. Epigenetics, it's all rooted in biochemistry. And there's plenty of other modifications shown in this table that I won't belabor. I would like you to notice how often nucleophilic residues are the ones that will get modified. And that's not a coincidence. Lots of amines and lots of alcohols. Another source of protein function resides within their cofactors. This typically refers to bound metal ions that are chelated by specific residues. For example, the acidic residues, aspartates and glutamates, these will prefer to bind so-called harder metal ions like calcium 2+. I think a great example of that is calsequestrin. You may remember that from physiology. It's a calcium sponge thanks to its plentiful D and E residues, and that protein helps induce the calcium-dependent contraction cycle within muscle tissue. Softer metal ions, which are metals that are farther down the periodic table, like copper-1, these will preferentially be chelated by sulfur-bearing residues. Cysteine, which has a thiol side chain, and methionine, which has a thioether side chain. Lastly, residues that contain an imidazole side group, specifically that would be histidine. These aren't too picky. They're happy to bind most metals, really. And the things that you can do with metals in biology is truly breathtaking. Electron transfer, for example, is something we will be discussing quite a bit later in the course. Water splitting is something that I find to be absolutely fascinating. It's the most important part of photosynthesis, in my opinion, and we will sadly not have time to talk about it here, but if you ever have the opportunity to take an advanced bioinorganic chemistry course, I highly recommend it. That is where you will get to see the full potential of biology and metals working together. Now, when an organic molecule binds to a protein and gives it some extra functionality, we call those coenzymes. Sometimes vitamins as well. Sometimes people call them cofactors too, even though they're not metals. I do know some scientists can get very particular about the semantics. I, for one, am not. I know prosthetic groups is another word used for coenzymes, particularly when the coenzyme doesn't really ever leave the protein. I think a great example of that would be heme. Heme is a coenzyme that we will be discussing very shortly, and it's really good at binding iron and sometimes even activating it if it's meant to be catalytic. Some other famous coenzymes that maybe you heard of: FAD, NAD+. These are derivatives of vitamins B2 and B3. We will be talking about these a lot in this course as well. They are important for hydride transfer, energy transfer. I would not call these prosthetic groups because they will pop on and off of enzymes very freely. And you can read through this table at your leisure because some of this we've already discussed or we will be discussing later on in more detail. I would like to comment on vitamin B12 really quickly just because I think it's cool. It's a very complicated looking vitamin. Its synthetic route took years to replicate in a lab setting and earned many people a Nobel Prize upon doing so. And what's really interesting about vitamin B12 is it's the only example of an organometallic bond, a carbon metal bond in biology. So I think that's pretty cool stuff. Now that we've talked about the many potential add-ons to proteins, let's begin dissecting their various functions. The transport/storage functionality has one of the most famous proteins as its poster child, and that would be hemoglobin. It's a dimer of dimer polypeptides found in red blood cells. It consists of two alpha chains and two beta chains stitched together, and their job is to ferry oxygen from the lungs to everywhere else. And hemoglobin can do that thanks to its very precise quaternary structure. That quaternary structure is stabilized through multiple hydrogen bonds, salt bridges, and of course the hydrophobic effect. And as a consequence, one hemoglobin protein will contain four heme prosthetic groups. And you can see them as these green molecules here, 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 here within the ribbon diagram. Down below, I have the skeletal structure, so you can take a look zoomed in. Do take note of the iron 2 atom that's sitting within the porphyrin ring. That iron is what will be binding dioxygen. And since hemoglobin has 4 hemes in it, we can at most carry 4 dioxygen molecules per protein. But it's not enough to just bind oxygen. Hemoglobin needs to deliver it safely too. And oxygen is pretty reactive on its own, and when it binds iron 2, it can go ballistic. A linear binding mode would very quickly convert dioxygen into the radical called superoxide, which is very dangerous. But hemoglobin is smart. That oxygen cavity in the hemoglobin fold is very awkwardly shaped. Oxygen can sneak on in, but it could only bind iron at a very uncomfortable angle. The nonlinear binding is what prevents iron from activating oxygen into superoxide, and that lets it be delivered safely to its final destination. Just how tightly does hemoglobin bind oxygen? If it's a tight bind, well, you may think that's a good thing, but how would hemoglobin then pry off the oxygen and deliver it to the tissues it circulates through? If it's a weak binder, how would hemoglobin sponge up oxygen in the first place? So the answer is yes. Ligands are in equilibrium, which means they come on and they come off. And how we describe if equilibrium favors binding or not is through the KD. You can sort of think of this as the acid dissociation constant equivalent to ligand binding. Specifically, it is the concentration of ligand, where 1/2 of all the binding sites become occupied or saturated. Said another way, a more mathematical way, the KD equals the product of dissociation products, so P for protein, free protein, times L for ligand, free ligand, all divided by the concentration of protein-ligand complex. Let's consider hemoglobin's oxygen binding curve shown in this isotherm on the right. And here, let's pay attention to the purple line. That corresponds to hemoglobin. What happens as we increase the concentration or the partial pressure of dissolved oxygen in blood on the x-axis? Hemoglobin's hemes will begin to saturate with oxygen. Well, in what manner? Saturation will be tracked on the y-axis, and indeed, as we increase dissolved oxygen, it will approach 100% saturation, but it will not do so linearly. It will instead do so sigmoidally. Pause the video and interpret this graph and predict what the KD would be for the oxygen hemoglobin system. I want you to recall that it's the concentration of ligand where half of the sites become occupied. So my strategy is to find the 50% mark and draw a line along in the x direction. Then take note where that line intersects the isotherm function. This should be here. From that point, drop on down to the x-axis and measure the reading. That to me looks like, I don't know, 25 torr. The KD for hemoglobin's binding isotherm. That is the amount of oxygen we need to get half maximal binding. Let's now compare this to hemoglobin's cousin, myoglobin, or MB for short. Myoglobin is also an oxygen storage protein, but this one instead is found in muscle. Okay? Its job is to take the oxygen from hemoglobin. And both proteins contain heme groups, but myoglobin will exist as a monomer, not a tetramer. Let's now compare the KD between hemoglobin and myoglobin. Again, pause the video, try to figure it out, and then follow along. We can use the same strategy as before, where we start at this 50% mark, and oh look, we cross the myoglobin isotherm, which is shown in blue, very early on. Drop down to the x-axis, and that looks like, to me, 5 torr. That is a substantially lower KD value. And you may ask, what is the significance of that? Does that mean more binding or less binding? Look at this equation to figure that out. If we had a very small KD, that means the concentration of protein-ligand complex is going to be way bigger than the numerator term, free protein and free ligand. So the smaller the KD is, the greater the affinity of that ligand will be. What's the advantage of that? Well, this differential in oxygen affinity helps drive migration from oxygen in the blood to oxygen-deprived tissues. Myoglobin likes oxygen more than hemoglobin. It wouldn't help our muscles if the blood loved oxygen so much it just held on to it forever. It would be like if the Amazon delivery person never actually gave you your packages. It would be thievery. And now look at this from hemoglobin's perspective again. Unlike myoglobin, it is not too keen to pick up oxygen when the ligand concentration is low, which is the case in our veins. That will change as the oxygen levels increase, and they will be very high in our arteries or blood coming straight from the lungs. Here, saturation is almost 100%. Look at this pattern. We have easy oxygen loading in the lungs and easy offloading at the extremities. Onload, offload. And that is all thanks to the sigmoidal binding pattern between oxygen and hemoglobin. Now, why isn't myoglobin's isotherm sigmoidal? This is a hyperbola. It's hyperbolic. Why isn't it just a stronger sigmoid, you may ask? The reason why is called cooperativity. Hemoglobin's quaternary structure makes its oxygen affinity increase as more subunits bind to it. And this is how. Deoxyhemoglobin naturally adopts a taut or tight configuration, and that configuration only modestly binds oxygen. Sometimes we also call it the T state. And you can see a depiction of that over here. And take note of the heme group. Notice how that porphyrin moiety in it is puckered, and that iron sits out of plane by 0.6 nanometers. It's not exactly housed in there nicely. Something dramatic is going to happen to this configuration when dioxygen enters that cavity and binds it. When that happens, we are going to have that oxygen pull the iron back into the plane of the heme group and flatten it out. And it's only going to tug on it 0.039 nanometers. That's how much of a shift we're getting to move this iron symmetrically within the plane of the porphyrin ring. That tug by the oxygen molecule doesn't just flatten out the heme, it's also going to tug this histidine residue by 0.039 nanometers as well in this direction. And it will also tug the alpha helix that the histidine is a part of by 0.039 nanometers. And that tiny, tiny adjustment actually will snap several salt bridges between an alpha and beta subunit. And breaking those quaternary contacts will not unravel the protein, but allow it to rearrange dramatically and have the alpha and beta subunits swivel by 15 degrees. That's a huge global rearrangement for such a small adjustment. And this swiveling converts the T state of hemoglobin into the R states, or the relaxed states. And when hemoglobin is relaxed, we reshape the oxygen cavities to allow for a greater affinity of O2 docking. And hopefully you can see in this cartoon that the more oxygens bind, the more relaxed the rest of the hemoglobin becomes. So to sum up, one tiny dioxygen molecule, 16 Daltons of mass, will bind 40,000 kilodaltons of protein and cause this massive rearrangement so necessary for life. Because again, in tissues where oxygen is abundant, like the lungs, we can use Le Chatelier's principle to drive the loading up of hemoglobin with oxygen past that initial tough bind. And in oxygen-starved tissues, it's easier for hemoglobin to unload because here we favor the dissociation of oxygen. Think of it this way, like trying broccoli. Getting over that first hump is difficult, but after having 2 or 3 pieces, the rest go down more easily. And this answers the question on the previous slide why hemoglobin has a sigmoidal curve, but myoglobin is hyperbolic. These adjustments, the T to R state, can only happen when you have quaternary structure. Myoglobin does not have quaternary structure, so there's no alpha and beta subunit swiveling that can happen. Hemoglobin is smart, and not only that, the body is smart too. It has learned to further regulate hemoglobin's oxygen binding allosterically. Tissues that work very hard, such as muscle, tend to produce a lot of acid. Their local pH tends to run a little on the low side. Let's check out hemoglobin's saturation curve as a function of acid added. We will start with this yellow curve, pH 7.6, and we will systematically lower it from the yellow, the green, to blue, all the way to the violet curve at pH 6.8. What happens to the Kd of hemoglobin in this experiment? As we increase the acidity, lower the pH, the Kd goes up. We started here, and we ended there. 25 to 55, it looks like. When Kd goes up, affinity for the ligand goes down. Hemoglobin loses affinity for oxygen in these acidic, hard-working environments. And that's not because the acid is denaturing the hemoglobin, quite the opposite. These lower pHs stabilize salt bridges that enforce hemoglobin's T state. And the T state is the configuration that's good for offloading oxygen. And this makes sense as hemoglobin passes through slightly acidic environments. Those environments need oxygen more, so it will preferentially drop off oxygen to those tissues. Furthermore, metabolically active tissues will also produce a lot of this compound called 2,3-bisphosphoglycerates. Here is the skeletal structure of that compound over here. It has many roles. It is a glycolysis stimulator on one hand. Not only that, it will bind hemoglobin's central cavity, which also stabilizes the T state. And let's think this through by consulting the isotherm again. In this graph, let's start with stripped hemoglobin, which is the green curve. It shoots up to 100% saturation quite quickly. When we add in bisphosphoglycerate, we're going to shift the curve and increase the KD. You may also notice other experiments were done here where we added in CO2, another compound released by actively respiring tissues, and that also shifts the KD upwards for hemoglobin. You put them both together and you get the most dramatic effect. All right? Again, it's the same kind of technology. Actively working cells will release compounds or acid, which will prompt hemoglobin to preferentially unload oxygen because those tissues are the most desperate for it. Alrighty, it's high time that we've discussed some other protein functionalities, not just limited to hemoglobin. Let's discuss recognition next. Recognition is the ability for an organism to detect specific substances. And this function is also built upon protein-ligand interactions. As a matter of fact, the best ligand binders in biology are antibodies, aka immunoglobulins. So when an organism is exposed to a foreign substance, which we call an antigen, and that stands for antibody generator, that organism's white blood cells will create unique immunoglobulins specifically tailored to bind and neutralize whatever that antigen is with extremely, extremely high affinity. The KD for a typical antibody is in the nanomolar range, so like 10 to the minus 9 molar. So you don't need a lot of ligand to drive complete binding. The structures of antibodies are quite various, but you can generally boil it down to the following. There's going to be sort of a common scaffold region that won't change too much. And the region that is variable can be customized to match the shape of whatever antigen you're on the hunt for. If you want to learn more about these, I highly recommend you take an advanced course on immunology. Another functionality that I'll just mention in passing is protein signaling. And in protein signaling, that's going to feature an external messenger, which is going to bind a receptor on the outside of the cell, and that binding will induce a response within the cell. Internal response. And I think a great example of that are receptor tyrosine kinases. This over here is a schematic of one type that's involved with growth factors. I don't expect you to know any fine details for this course, but basically a growth factor is a protein signaler that will instruct a cell to multiply. It's going to bind and dimerize a specific receptor tyrosine kinase protein, and that's going to trigger all sorts of intricate intracellular machinery that will ultimately change which proteins get transcribed in the cell nucleus.
And the new proteins that get created in response to this growth factor binding are going to instruct and prepare the cell to divide, for example. And depending on what this signaler is and what receptor it binds, you can get a whole sort of other kinds of physiological responses. If you want to learn more about how protein signaling works, then I highly recommend you take cell biology or cancer biology if you want to know what happens when these signaling pathways don't work or work maybe a little bit too well. The last kind of protein function I'll introduce will be the focus for the next several lectures, and that's catalysis, enzymes. The proteins that catalyze life's many reactions. The part of an enzyme that catalyzes reactions specifically is called the active site. And the active site technically has two subcomponents. The binding site, which, as the name suggests, binds the substrate, and the catalytic site, which actually does the chemistry upon the substrate. This can be a little bit pedantic because sometimes these are the same thing, often they're not. I know a lot of chemists that use these terms interchangeably. Sometimes I do so, but technically there is a difference between the active site, the binding site, and the catalytic site, even though it's very, very subtle. More interesting than definitions, in my opinion, are the many, many types of enzymes out there. But believe it or not, of the hundreds of thousands, if not millions, of enzymes in existence, they actually only perform 7 types of general reactions. Oxidoreductases, you'll see a bunch of these, as the name suggests, will either oxidize or reduce a substrate, usually through this process called hydride transfer. We'll talk more about that soon enough. We also have transferases, and what these will do is transfer a functional group from one molecule to another. And you can sort of just go through these on your own time. I'm not going to read them out to you one by one. The only thing I'm going to sort of mention is maybe the lyases, because they're the only class of enzyme that's not intuitive from the construction of the name. And basically, what a lyase does will form or break carbon-carbon bonds or carbon-oxygen, carbon-nitrogen bonds through a variety of specific mechanisms, eliminations, for example, or addition reactions. They usually don't involve simple substitutions. You may also notice that all of these class names end with the suffix ase, oxidoreductase, isomerase, ligase, so on and so forth. That's usually a good clue that this protein, whatever it is some kind of enzyme. The model that explains why enzymes are so good at speeding up reactions is called the induced fit model. I like to think of this as the hand and glove model. All right. Like wiggling your hand into a glove, the natural stretching and wiggling of the protein in solution is going to allow its substrate to worm into the binding pocket. And then these substrates are going to be manipulated in the glove to react together thanks to these flexible enzyme motions or arranging them in very specific ways. Sometimes we call this breathing motions. And this is something I like to emphasize in this course because up until now, you may have been taught that enzymes bind their substrates in this lock and key fashion, which is very specific and rigid. And that's not reality at all, really. It is specific and flexible, hence induced fit model. Now, in every enzyme, there are 4 general strategies that they use to get their substrates to react. And I'm going to take you through them one by one. For starters, the first strategy is called catalysis by approximation. This is where substrate proximity is going to drive reactivity. What does that mean in plain English? In this enzyme schematic shown over here, there are 2 binding pockets that are shown in blue. We're going to dock this hexagon over here into that binding pocket, and this rectangle over there in that binding pocket. Once we dock, the enzyme, kind of like in the case with oxygen and hemoglobin, is going to undergo a dramatic global rearrangement. These 2 lobes are going to come together and kind of wrap around the substrate a little bit. It's going to, well, put a little bit of mechanical stress on the substrates for starters, but also it's going to position them in just the right way where they can react with each other at the catalytic site in between. Think of it this way. The chance of this rectangle and this hexagon colliding with each other in solution at just the right speed and just the right orientation to build a hypothetical bond between them, let's say, doing that just right randomly is really hard. There's going to be a very low chance of that happening. The entropic barrier is high. But what the enzyme does is reduce the entropic barrier of these coming together in just the right way. And that is what speeds up the reaction. We can facilitate this bond forming without the enzyme even getting its hand dirty, just by bringing 2 things together inside of its pockets. Now, don't be tempted to think that the gray region highlighted there is useless. Without that gray stuff, there's no way for the enzyme to respond to the substrate binding event like so. There's no way to form this enzyme substrate complex that is active. So just because there are parts of the protein that are not catalytic does not mean that those parts are useless. The second catalytic strategy I'm going to discuss is called acid-base catalysis. Here, the enzyme is going to reversibly protonate and or deprotonate the substrate via its acidic and basic residues. And it will do that quite often because that's usually a prerequisite to many types of organic reactions. And to illustrate what I mean by that, let's take a look at this figure over here. We have sort of a generic nucleophilic attack being described when this hydroxyl takes its lone pairs and does a backside attack on this carbonyl carbon. So this looks like an SN2 process, right? And we have an electrophile and a nucleophile. In this case, the hydroxyl is doing the attacking, so it's nucleophilic. And the victim, the attackee, is the electrophile.
Now, this isn't a terrific reaction because hydroxyls generally are rather poor nucleophiles, and we can look at the alleged products to see why. When we form a bond between this nucleophile and that electrophile, we get this COC positive entity called an oxonium, which is very unstable. So this reaction is not really favored as is and doesn't actually happen. However, how can the enzyme facilitate this bond-forming reaction? Through a little bit of acid-base catalysis. What if we had a residue, a basic residue, that deprotonated and stole that hydrogen right off of the hydroxyl group? What's the consequence of that? We're going to go from a neutral OH to a negatively charged alkoxide, O-. And alkoxides are way more nucleophilic, way more reactive than their hydroxyl counterparts. And this is why. When that O- attacks the electrophilic ketone carbon, what we will form instead is a neutral COC, and that's way more favorable, way more stable, and in actuality is what happens. Now, that's not the only instance of acid-base catalysis that's occurring here. We need to think about the leaving group next. Let's go back to this diagram and see what happens in the uncatalyzed case. When we form this bond between the oxygen and the carbon, we can't forget about the negative charge, that lone pair that got kicked up in this part of the mechanism. That negative charge is going to swing back down, and we're going to reform this C double bond O that was initially present. But we can't have 5 bonds to that central carbon. One of those other 3 bonds needs to break. So in the case of this reaction, it's really easy to break a CO positive bond. So that's why that reaction doesn't really occur. However, if we stabilize that by getting rid of this H and that charge, well, which bond do we cut then? The CO neutral bond or the CN neutral bond? Is there a way we can make this decision a little bit easier? Well, if we had a general acid residue, something with a proton to spare, it could deliver that proton to that nitrogen, which will protonate it and give it a positive charge, like so. So let's jump down here and see what we get when we do that. We have this NHH plus, and this is a really good leaving group. That N plus is like an electron vacuum, and so it's going to slurp up the electrons in that bond as a lone pair, causing it to break and leave. So by having general acid and general base catalysis working in tandem, we can make something a good nucleophile and make another part of the molecule a good leaving group, therefore allowing this reaction to proceed as such. So this does happen. The third strategy, covalent catalysis, is a little similar to acid-base catalysis, except here the enzyme will form, again, a reversible covalent bond
with the substrate. And the goal here is to lock it in temporarily. A very common mechanism for covalent catalysis occurs when you have a reactive lysine, which has an NH2 side chain, and that's going to nucleophilically attack a substrate ketone carbon, and that is going to react in the following manner. We're going to form a bond and then dehydrate this OH equivalent to form something that's known as the Schiff base. This is a somewhat stable bond between the enzyme itself and the substrate intermediate that we can easily break later on once we're done reacting with that substrate, simply by adding water back to the bond, which will cleave it, therefore releasing the product and allowing the enzyme to continue its reaction cycles. I need to emphasize here that even though the enzyme is technically reacting with the substrate in both of these cases, neither instance is permanent. When all is said and done, the substrate will leave as a product and the enzyme will be regenerated as it was in its original state. The enzyme is going to be an active participant in the reaction mechanism, but once it's complete, it will ultimately not be changed at all. Metal ions have a functional role in about half of all known proteins, and in enzymes, they enable the final strategy of catalysis, which is metal ion catalysis. And this has many forms. Metal ions, for example, can help a reaction occur by positioning substrates into a configuration that is ripe for reacting. Another word for this is catalysis by approximation, right? We are lowering the entropy, we're ordering the substrates, and that is going to help them react in the way they're meant to. In addition to that, metal ion catalysis can also stabilize high energy intermediates and transition states, which often bear unstable charges. And a way that we can illustrate both of these processes working together is in this snippet of the mechanism for the enzyme thermolysin. Thermolysin is an example of a hydrolase. We mentioned hydrolases a few slides back. And hydrolases in general will activate a water molecule and use it to break apart a bond. So in the case of thermolysin, the nucleophile is going to be water, and the electrophile is this amide carbon in this amide bond shown over there. Now, water and amides typically don't react with each other very easily, but what this common zinc cofactor is doing is it's forming a dative bond, it's called, between the metal ion and these two oxygens, and that's going to help bring them together and put them into a reacting proximity. But we need a little bit more than that to get this reaction going, because water is a terrible nucleophile. So this nearby glutamic acid residue is going to do acid-base catalysis and deprotonate the water into hydroxide, O-. That's a great nucleophile. And that is going to very easily form a bond with that electrophilic carbon. Now, once we do that, this pi bond is going to be kicked up onto that oxygen as a lone pair, establishing a discrete negative charge. That's a little bit unstable, but remember we have our zinc cofactor here to save the day. Its 2+ charge is able to stabilize that O- and help it form
so we can then proceed with the rest of the mechanism and then ultimately clip off this amide bond. So there you have it, metal ion catalysis, positioning two substrates together and then stabilizing the intermediate that forms once they react. Now, there's actually a third way that metal ions can facilitate catalysis, and that's where the metal itself becomes reactive and drives redox reactions by changing its oxidation state. Metal ions are really good at doing redox, and we can see an example of that in this mechanism for a class of enzymes known as cytochromes. Now, cytochromes usually feature a iron cofactor, and by changing its oxidation state from iron 3 to iron 2 to iron 5, we're able to do all sorts of crazy chemistry. And we can, for example, activate an oxygen molecule and a carbon-hydrogen bond, which is normally not redox active, but hey, we can oxidize it with that oxygen to form COH, a hydroxyl group. It is really difficult to break a carbon-hydrogen bond, and really the best way of doing that in a lot of cases is to use something like a cytochrome. Now, I don't expect you to fully understand or memorize this mechanism. This is some really advanced stuff. What I want you to take away here is that the metal ion is able to do redox reactions upon organic substrates. The cool thing about enzymes, all proteins really, is that we haven't even discovered half of them yet. The sky is really the limit here. I want you to consider DCE, for example. That is an environmental pollutant that is generated from PVC pipe degradation. This slowly over time gets leached out of the pipes and into the water supply. And this is, a thing of environmental concern. But some clever scientists, for example, were able to address this concern by mutating in the lab an enzyme found in bacteria called epoxide hydrolase. So by using some human ingenuity, we can re-engineer this enzyme so that it specifically targets DCE, and it's able to break it down in a two-step process into a much less toxic substrate called glyoxal. And so that's an example of how we can take already existing proteins and fine-tune them to address real-world problems. And I think this is pretty cool as well. In 2016, we actually discovered that a kind of bacterium that was in a polluted lake outside a water bottle factory did this chemistry all on its own. There was no scientists involved. The bacterium evolved and made a brand new enzyme from scratch called PETase. This enzyme has the ability to break down and digest plastic, something that industry has not found an easy way of doing, and yet biology figured it out in only a few decades after the birth of plastics. I mean, these bacteria were minding their own business in a lake, plastic waste began accumulating in it, and they figured a way to turn that into an advantage for them. And these are just two very small specific examples. Are today's problems going to be solved with tomorrow's proteins? That is a question that only you can answer.